Welcome back, ladies and gentlemen. So today we're really going to start talking about the fact that there's a spectrum to dominance. When we talk about Mendelian genetics, it really is either it's purple or white, tall or short, wrinkled or smooth. And yet most of the characteristics we look at in organisms don't really work quite that simplistically. Um, because what we're really looking at a molecular level is that those dominant and recessive alleles aren't truly interacting with each other. What's happening is that you have a sequence of A's, T's, C's, and G's that lead to that quote unquote dominant protein. Recessive is a different sequence that then leads to the production of a different protein. And what's really happening is the synthesis of those two proteins and the way that they interact with each other is what drives the phenotype of the organism. So when we look at an individual who is heterozygous under that sort of traditional Mendelian genetics, they have the code for that purple flower as well as the code for that white flower. And they are producing both the purple and the white proteins, but it's then a question of what does the expression look like when both of those proteins are present. That's what then determines the phenotype. So we can have a huge range of dominance as a result of those heterozygotes because you've got both protein codes and as a result both proteins being produced. The other thing to keep in mind here when we're talking about dominance is that dominant alleles don't necessarily equate to being more common. That dominant allele simply states that in traditional Mendelian genetics, the presence of that protein overshadows the capacity for you to physically see the functionality of the recessive protein in a heterozygote individual. There are plenty of characteristics that are recessive that are actually more common. Take a look down at your hands. All right, I'm going to assume that the vast majority of you sitting out there right now don't have webbing between your fingers. And yet, that lack of webbing is a recessive characteristic, not a dominant one. So, dominance, don't be confusing that with meaning common. It just has to do with the production of those proteins and how they interact with each other when both allele sequences are present.
All right, guys, so welcome back. We're going to pick up our discussion on genetics by talking about the fact that while Mendel's experiments and looking at things like his pea plants were incredibly revolutionary at the time, they were also something that he was incredibly lucky to have figured out. He picked a handful of traits from an organism that really worked in that very simplistic, it's either this or that. Very contrasting traits that happen to be inherited that way. That's how he was able to discover dominant versus recessive, heterozygous, homozygous, coming up with all of those hypotheses that are fundamental to our understanding of Mendelian genetics. But here's the thing, when we start really looking at the full gamut of characteristics in living organisms, overwhelmingly, most of them are not anywhere near as simple an inheritance pattern as Mendel saw with those pea plants. So when we look at inheritance of a character, even if it is by a single gene, there are a lot of situations where those alleles are either not truly dominant or recessive. There's some in between. Some genes, although it's a single trait, there are multiple alleles. So it's not just purple versus white. There are other variations in between. And in other cases, there are genes that produce multiple phenotypes because of the way that these genes work. So keep in mind when we're talking about genetics, we're talking about the phenotypic expression of a set of instructions. So your DNA has a particular A, T, C, and G code for that gene that goes then through transcription and translation to ultimately produce a protein in your cells. We're also talking about organisms that are diploid, so they have two sets or two copies of every allele, every gene. So the allele they get from their mother is expressing one protein, the information from their father may be expressing the same protein and it may be expressing something else. So the interaction of those proteins in the cells all plays into how that phenotype, that physical expression of that genetic information ultimately takes place. So when we look at a dominant versus recessive allele. Remember the way that Mendel describes this. So we talk about your homozygous, heterozygous, and then homozygous dominant versus homozygous recessive, right? So in the way that all of Mendel's traits worked, you had that homozygous dominant who had two copies, one copy from each parent. Both of those genes ultimately expressed the same protein, and so you saw that particular color, for example. Heterozygous, you got now two contrasting alleles, so a dominant and a recessive, but in Mendel's work, that dominant trait, the protein that it produced, ultimately masked the fact that that recessive protein was still being produced. So in a heterozygote, you're still getting those two different proteins being produced by the cell, but in Mendel's work, the dominant overshadowed the expression of that recessive protein. You go to a homozygous recessive, now you got two copies of that recessive protein being produced, and so you saw the recessive phenotype. But when we look at most of the traits in organisms, things like Tay-Sachs disease, for example, depending on even the level at which we study that particular characteristic, the inheritance pattern may not necessarily be cut and dry. It may not necessarily be identical at every level. So most of what we focus on, especially for students learning genetics as underclassmen, so your freshman, sophomore bio courses, would have looked and focused mostly on this organism level. What is the DNA expressing that we ultimately see as the function of the whole organism? So something like Tay-Sachs disease, which is a fetal disease that ultimately results in an enzyme in your brain that is not doing its job appropriately, and as a result causes this accumulation of lipids in the brain. At an organism level, if I study it there and look at how it gets passed from parent to offspring and how it gets passed from generation to generation throughout families, it appears to be an overall recessively inherited trait. That being said, if I go down to the biochemical level and actually look at how that phenotype enzyme activity level is being expressed, it's actually incompletely dominant. And if I go all the way down to the molecular level and actually look at the protein production, it's actually codominant. So when you talk about this spectrum of dominance, you also have to include at which level are you focusing on the expression of that genetic information. Because here's a great example where Mendel was working at the organism level. And so this would have looked much more cut and dry, like a dominant versus recessive trait. But as we have gained more understanding of biochemistry and being able to dig into more of that cellular biology that he was not necessarily aware of, we realize that it's not necessarily inherited in the same way, expressed in the same way at every level of an organism's function. So just kind of tuck that in the back of your head and keep that in mind. In addition to that, I mentioned things like codominance and things like that. There are a huge range of ways that characteristics that are controlled even by a single gene deviate from those simple dominant versus recessive traits that Mendel saw in his pea plants. And so this is the list of characteristics, alternative inheritance patterns that we're going to kind of focus on today.